All right, well, let's get going with chapter 5, shall we? I think we're going to break this into maybe two parts here, but this topic is now that we have finished talking about action potentials in the previous chapter and conduction of the nerve impulse along the axon. What happens at the end of the axon? This is when a neuron communicates with another neuron at the synapse. So we need to know some things about synaptic transmission at this point. And to talk about that, we can come back and revisit the discoveries of Santiago Ramon y Cajal. If you remember, he was the one who, using the newly developed stain invented by Samuel Golgi, and was able to visualize neurons under a microscope, and he produced a number of remarkable drawings of neurons. But one of the things that he found in his observations is that the nerves that he was viewing were not long continuous fibers, but consisted of these discrete individual cells that had small gaps between them. And so this does raise the question of how do neurons communicate with each other, assuming that they do at this point, which at that point in the history of this was pretty much a hypothesis, really. How does that work, right? And it was later that Sir Charles Sherrington discovered the nature of these communication systems between the neurons. And Sherrington was the one who actually gave us the word synapse. He also gave us a lot of other words that we use in psychology, including the word proprioception, which is our body's ability to detect touch and internal physical stimulation, right? Not just of our sense of touch, but also receptors that are embedded in muscles and tendons that provide us information about the movement and position of our joints. And when Sherrington was studying reflexive actions here in dogs, to understand the nature of synaptic function, he realized that there had to be receptors embedded particularly within their tendons in order to activate what's known as the tendon stretch reflex. The tendon stretch reflex is essentially the same thing that occurs in the stereotypical case of a doctor tapping your knee with a small hammer. What happens is that the hammer blow to just the right location in your knee stretches a tendon, and that signal transmits to your spine. And that stretching of the tendon activates a sensory neuron within the tendon. That signal sends an axon potential and a nerve impulse to the spine, where it directly synapses onto a motor neuron, which triggers a stretch response out of a knee flexor muscle, right? And so as the knee flexor muscle is activated, that muscle contracts, and that causes that little jerky kicking motion of the knee. So as you can imagine here, that entails a sensory neuron that has to be embedded into the knee tendon. It travels to the spine and it synapses onto a motor neuron, which triggers a muscle contraction. So two neurons are all that are involved in the simple version of this reflex. Now we're going to see that there are some other variations that can occur here in the reflex that might involve a couple of more neurons. But one of course the important things to talk about is that reflexes like this don't really involve the brain. And so let's first visualize what's happening here. So here's a picture that shows something like the tendon stretch reflex. We have a sensory neuron. Here it's shown as being embedded in the skin, but we could also imagine this again, as Sherrington had hypothesized, could also be embedded into a tendon within the knee. And that's going to send a sensory signal up to the spine. So remember, we had previously talked about the anatomy of the spine and argued that there were two different nerve roots that connect to the spine. So here's a spinal nerve that is connecting to the spine, but just before it arrives to the spine, it breaks into two roots. The dorsal root enters into the back of the spine, and the ventral root enters into the front or belly side of the spine. And the research that we had talked about previously of Charles Bell and Francois Magendie tells us that they've figured out that the dorsal root carries the sensory information or the afferents into the spine from the sensory receptor, which is then further transmitted to the brain, right? And also the ventral root is the motor signal that goes from the spine, a motor neuron in the spine, out to the muscles, synapsing directly onto a muscle fiber to cause muscle contraction. And so, of course, conscious voluntary motor control can also work by a descending signal that comes from the brain, as we see a brain neuron also shown in this diagram that would descend down to this part of the spine. And that brain neuron could be part of the motor cortex, in which it's sending a signal down to the motor neurons to trigger muscle contractions, or it could be in the somatosensory cortex, where it is receiving sensory input and we develop some conscious perception of any stimulation of the body. But as I was noting, these brain neurons are not really involved in any reflexive action that occurs at the knee, in the knee jerk reflex, right? That sensory motor neuron, the sensory neuron would directly synapse onto the motor neuron. And here we're seeing something like that, although there's something else happening here. There is something else labeled an intrinsic neuron. An intrinsic neuron is a neuron that just is there within the spine, and it can communicate with that motor neuron, or it can communicate with some other neurons elsewhere in the spine, or it can communicate with the brain, right? And so this is an example of what we had previously called interneurons, right? So when we talked about different kinds of neurons, we talked about sensory neurons, motor neurons, but also interneurons are neurons that communicate between other neurons. And so this intrinsic neuron is called intrinsic because it exists intrinsically within the spinal cord itself, but it's also an example of an interneuron that we have previously labeled. So let's talk about Sherrington's discoveries. And I have 4 of them here. And I'm going to provide a little bit more detail as I jump forward to look at some of these other slides coming up and a few pictures that I have for this. Number 1, Sherrington was interested in studying the speed of the nerve impulse. And we already had some stats on how fast a nerve impulse can travel in the previous chapter. One question we might want to ask is, how do we know how fast a nerve impulse can travel? One way to do this is to look at reaction time studies. And this is something that was actually pioneered prior to Sherrington by a guy named Hermann von Helmholtz. And one of the things that Helmholtz would do is he would attach a stimulus to a person's knee, right? And this could be something like an electrical shock or some other painful stimulus. And once he applies the electrical shock, the person's job in this study is to press a button as fast as they can, which will turn the shock off, right? So it's very clearly a reaction time task here. As soon as you feel that shock happen to your knee, push down this key, which will deactivate that shock. But this was hooked up to a timer so that Helmholtz was able to calculate the reaction time to that stimulus. But what Helmholtz had also done was he redid this experiment, but he applied the electrical shock to a person's heel instead of their knee. And what he found is that the reaction time in that case was a little bit longer compared to when the stimulus was at the knee. And his explanation for this is that the nerve impulse, once that shock stimulus is applied at the heel, has to travel a further distance to get to the spine, and then a further up to the brain to be recognized and detected so that the reaction can be activated. And so the nerve impulse has to travel a little bit farther, right? The distance, the extra distance between the two is the distance between the person's knee and their heel. And if you take that distance, which you could measure simply enough with a ruler, and divide that distance by the difference in the reaction times of those two tasks, you could have a distance per time calculation, which is, of course, exactly what a velocity measurement is. It's a certain number of, say, meters per second, right? But one of the things that Sherrington did in studying reaction times to various stimuli and looking at the estimating, like Helmholtz was doing, estimating the speed of a nerve impulse, is that he found that if you have extra synapses involved in a reaction time task, the the reaction time is a little bit slower. Let me jump ahead to this real quick. So let's look at discovery number 1, speed of the synaptic transmission. So based on Sherrington's refinements of Helmholtz techniques, he estimated that the speed of a nerve impulse along an axon is 40 meters per second. That's pretty quick, right? But one of the things that Sherrington had noted is that when studying certain reflexive actions, and looking at the reaction time to activate a reflexive response, just like the knee jerk, right? So the question here is, let's tap the knee tendon, and then wait for that kicking response. And we're going to measure the delay, and that we're going to call that the response latency. And then we're going to measure the distance from the point of contact on the knee where the stimulus was applied to the spine, and then from the spine back to the muscle that contracts. And we can kind of figure out what the total distance the nerve impulse has to be is. And then we can measure the reaction time in that task. And then again, we'll calculate sort of a distance over time, and that'll give us a velocity measurement here. In this case, he found that the velocity was significantly slower, right? Around 15 meters per second. And Sherrington's claim here is that the reason this is so much slower compared to just pure transmission along a single nerve axon is that the synapse has to be there to transmit that action potential from one cell to another. And that synaptic transmission is slower than the nerve impulse down the axon. And so this was the initial observation that Sherrington had made that kind of proved the existence of the synapse, right? Separate from Ramon Cajal's observation of these small gaps, Sherrington is now arguing that when you have communication across a synapse, that is slower than communication down an axon, and the presence of a synapse can be demonstrated whenever we see that kind of slowing of a response. So I have another picture here real quick, fairly simple little diagram that just shows the basic idea that if we're going to have neuron A sending a signal to neuron B, we can measure the duration or the speed of that signal down the axon of neuron A as 40 meters per second. But if we're going to have a signal go from neuron C to neuron E, but we have an interneuron D in between, and we have an extra synapse here, right? So in the top of this, we have just one synapse, but in the bottom, we have two synapses. This is when the reaction time between C and E is a lot slower, and that can only be explained by the presence of this extra synapse between C and D. And that has such an effect here that delays the signal so much that this is what Sherrington had used to argue that synaptic transmission is a lot slower than nerve transmission, but of course, merely demonstrates the existence of the synapse as something that is part of sending a signal in the nervous system. Now, let me go back to the earlier slide here where I had this list of his discoveries. A second discovery from Sherrington is called temporal summation. And as I have it written out here, several weak stimuli presented at different times can combine themselves together to create a stronger response than any single stimulus. So what does that mean? Let's go to the slide on that, which is right here. So again, let's imagine trying to do something to activate the knee jerk reflex. And we would imagine that in order to activate the knee jerk reflex, we're going to strike the knee with a hammer. Again, just a little rubber mallet, right? Not anything super painful. But we have to strike the knee with that rubber mallet sufficiently hard enough to activate a signal in the sensory neuron embedded into that knee tendon, right? And if we don't strike the knee tendon strong enough, it's not going to stimulate that neuron. Right? And remember what we had learned about with neurons and action potentials in the previous chapter is that neurons have what's called a firing threshold. They have a resting state of -70 millivolts, but in order to activate the action potential, we have to depolarize that neuron at least to the point where it rises its internal voltage above the firing threshold, which is typically something like negative 50 millivolts, right? So we have to raise it by about 20 millivolts. So a very soft tap with our rubber mallet might raise the voltage of that cell a little bit, but it might take us from negative 70 to negative 60, right? Which is not enough to cause that neuron to actually fire an action potential and transmit a signal to the spine where it can activate that motor neuron and trigger the actual knee-jerk reflex, right? So we have to strike that neuron strongly enough to activate the response. But what Sherrington found is that if I apply a soft tap that individually is not strong enough to activate the reflex, but I very quickly apply a second soft tap, those two soft taps together can combine their influence on the sensory receptor so that now we actually get a reaction here, right? So even though the individual strength of any one of those taps is too weak to activate the neuron, two successive taps that are weak like that add their effects together, right? And that's why we're calling this summation, because these two different stimuli can sum together their influence onto the neuron. And in this particular case, we're going to call it temporal summation because these two stimuli are occurring at different times. There's a very brief delay between them. Now, obviously, there can't be a really long delay, right? In fact, let's take a look at that. Let's come to this picture right here. There's a term on this image that we haven't really talked about yet, and we're going to find a little more detail later in the notes, but let me briefly explain what is an EPSP. That stands for an excitatory postsynaptic potential. And what we're talking about here is the idea is that the signal activating on the postsynaptic neuron. So the postsynaptic neuron is the neuron that's receiving the stimulus. And this is an excitatory stimulus. So it's going to trigger, if it's strong enough, at least, it's going to trigger excitation of the neuron, and it's going to trigger a depolarization. We're raising the internal voltage, right? And we see in the point labeled 1 here, a very small EPSP. And these are called, as you might have saw it on the previous slide, a graded potential. That is a very slight change, a graded slight change in the internal voltage of the cell. But at location 1 in this graph, that little blip of an EPSP is only about 5 millivolts. So it takes us from negative 70 to maybe negative 65. If nothing else happens, then that's not enough to cause an action potential. So the sodium-potassium pump is just going to do its job, and it's going to return this neuron back to its resting potential of negative 70. Now, if we have two stimuli that have this kind of an effect on the neuron, and they occur close enough together in time. So you can see in part 1 here that it takes a small amount of time for that EPSP or that graded potential to dissipate and for the neuron to return back to its resting state. So what if halfway through that little spike, we apply a second stimulus, and now that raises us from the negative 65 up to maybe negative 55? Again, that's not enough to activate this neuron because we still haven't crossed the firing threshold. But now at point 3 on this graph, what if we combine 3 stimuli? So we get 3 excitatory stimulations here, 3 graded potentials, and together, because they are slightly delayed, so they're still overlapping each other a little bit here, they add together their effects, and now we have hit that negative 50 activation threshold. And now we're going to see a typical action potential occurring in that neuron, right? So in that case, any one individual stimulus is too weak to stimulate that particular neuron. But if those 3 weak stimuli occur close enough together in time, they're going to combine their effects, right? And let's come back. That is called temporal summation. Weak stimuli create graded potentials. These will fade away, but if we apply a second or even a third graded potential within a rapid enough succession, they get to sum together their effects. Those graded potentials add together, and we end up with an actual action potential occurring, right? We're going to depolarize the neuron and see an action potential. And so that's temporal summation. So again, this is how Sherrington did it, right? He's trying to activate the reflex in dogs by applying a very weak stimulus that by itself cannot activate a reflex. But if he applies 2 or 3 of these weak stimuli in very rapid succession, boom, there you get the reflex. So he's demonstrated now temporal summation. 3 stimuli that occur at different times, sum together, and that combination now activates the reflex. Let's look at discovery 3. Let me go back to my original list, which was here. A third discovery is called spatial summation. Stimuli at different locations produce a stronger reflex than at a single stimulus. So let's think again, this idea that if I apply a single stimulus that is not strong enough to activate a single reaction, but if I apply 3 different stimuli at 3 different locations on the body, maybe that will do the job. So let's go back to my text right here. So again, we're trying to activate a reflex. We apply a weak stimulus. That is not strong enough to activate the reflex. But if I apply several weak stimuli all at the same time, so now this is different than the other experiment, because in the previous experiment with temporal summation, we're applying several weak stimuli at slight delays from each other. But here we're going to apply them all simultaneously, but at different locations on the body. And what's going to happen, though, is the idea is that they all still converge to the same receptor. And therefore, those 3 different incoming signals all arrive at the same time from different locations to the same receptor. And each one is producing, again, a graded potential, but they get to add together or sum their combined effects to create a strong enough signal. And again, we end up with an action potential, and we're going to call this spatial summation. Let's go back to that other graph. So the point 4 here is showing us what we might see with a spatial summation situation here. So several different EPSPs all occurring at the same time, and even though individually they're all weak, as in point number 1 on this graph, if they all occur at the same time, they get to add together their effects simultaneously, and we see that rapid ramping up of the internal voltage of the cell enough to trigger the action potential. One thing to say about this, here's another picture, by the way, which helps us maybe visualize how temporal and spatial summation work in an anatomical way. So here we have a neuron. We're going to call this neuron on the left the presynaptic cell because it's receiving input, and we want to trigger an action potential in this cell that's going to travel to another neuron over on the right-hand side, which is the postsynaptic neuron. But how do we activate an action potential in this presynaptic neuron here? Well, on the top, we see 3 different arrows coming into a dendrite here, and they are coming in sequences, right? So they're coming together in a fast enough sequence that together they can still add their effects together and stimulate an action potential here. So we're going to get temporal summation. But on the bottom here, we're seeing 2 different signals coming in through 2 different axons, maybe coming from 2 different other neurons, arriving at 2 different dendrites, but they're coming in at the same time. And so together, the graded potential from those 2 separate signals is enough to stimulate an action potential into this neuron. And so we're going to call that now spatial summation, because we're summing across spatial positions of receptors on the skin surface here, as opposed to one position. In temporal summation, it's the same position on the skin, but at different points in time. In spatial summation, it's different locations on the skin separated by some spatial position. But one thing I want to say real quick about the issue of spatial summation, this is not quite what Sherrington was doing, but it's something that commonly occurs in the case of vision. If you've had the sensation and perception class, you might have learned about rods and cones. And one of the things you would learn about rod vision is that rods work very well in low light conditions. So when you're in a dark room, like you've been sitting in a dark room for 30 minutes and you are what we call fully dark adapted, you have switched to using rod vision instead of your cones. Your cones simply can't function well in darkness. But why is it that your rods are so much better at seeing in the dark compared to the cones? Because your rods use spatial summation and the cones do not. What does that mean? So imagine there's a an area on the back of your eye, the retina, where all of these light-sensitive cells are found, and you have a distribution of light across that area of the retina, but that light is very, very dim, right? So individually, each rod is receiving a very, very faint stimulus, right? And that means that these rods, and when they synapse onto some other neurons that are sending a signal to the brain, these are called ganglion cells in the retina, those individual rods are sending a graded potential. So they're all simultaneously activated by this very, very weak light. But because many rods at different locations on the retina are converging to the same ganglion cell, they are combining those graded potentials to stimulate that ganglion cell. And so the fact that that ganglion cell is now activated by all of these many rods that are simultaneously converging onto it, that's a signal that gets sent to the brain, and the brain can then say, oh, I can see, because that light was enough to trigger a nerve impulse that goes from the rods to the ganglion cells and on back to the brain. But in the case of the cones, because they're not really converging like that onto ganglion cells, they don't do spatial summation. Each individual cone is sending a weak graded potential to its corresponding ganglion cell, but that ganglion cell receiving that graded potential is not being activated, right? It's not enough to cross its firing threshold. So that ganglion cell does not send a signal to the brain. And therefore, the brain says, the cones are not telling me anything. The cones are not functioning at all, but the rods are. And so we're going to use the rod system to see through that spatial summation process. We can use the rods to see in low light because of the way in which they're using spatial summation to combine their effects to communicate with some of these other neurons that actually send the signals to the brain. But let's move on. There is one more thing. Let's go back to my original list again. Sorry about the jumping all around here, but we've covered temporal and spatial summation. So point number 4 of Sherrington's discovery is that he discovered that synapses can be excitatory, and that's what we've been talking about so far, these graded potentials adding up to create enough to excite the neuron and trigger an action potential. Those EPSPs that I had talked about, those are excitatory postsynaptic potentials. But Sherrington has also discovered inhibitory postsynaptic potentials, or IPSPs, and that was also in, where was it? Here we go. This particular image, number 5. We see what happens with an inhibitory signal is that instead of raising the internal voltage closer to the threshold, and if we cross that threshold, we're going to trigger an action potential. Now what we're going to do, and that's called, if you remember, it's called depolarization from the previous chapter. In this case, what we are doing is we are hyperpolarizing the neuron. And that means we are taking that polarized state of -70 millivolts and we're increasing it even further below 0, right? And they're going to take it down to -75 or -80, right? And that's what an IPSP does, right? So let's look at this now. So let's find where, well, I have more detail here, actually, before I get to the IPSP experiment that Sherrington had done. So again, some of the language of this is that we can talk about presynaptic neurons and postsynaptic neurons. So this is some basic vocabulary here. And so the excitatory postsynaptic potential, or the EPSP, is that graded potential that decays over time or space, right? But if we add enough of those together, we get the actual response. And let's talk about the inhibitory effect that Sherrington found. So here, Sherrington was doing something else. He was applying a painful stimulus to a dog's leg, just pinching it, and nothing terrible here. But what the dog would do is flex that leg, right? In response to that painful stimulus, the dog is going to flex the leg to withdraw and move it away, right? But he's also monitoring the muscles in the dog's leg here. And one of the things that he notes is that to control movement at a joint, there are 2 different muscles that work in opposition to each other. We call these agonist and antagonist muscles. Let's talk about the human arm for a second. Right? I don't know if you can see my arm here, right? So let's get my arm out, right? Not much of a muscle there, but it's there. So I'm going to move my elbow, right? In order to bring my elbow and my forearm closer to my head, I have to use the bicep muscle for that. I'm going to shorten and contract the bicep muscle for that. At the same time, on the back side of my arm, we have what is known as the tricep muscle, right? And I don't want the tricep muscle to be active here while I'm trying to use the bicep, right, to bring my forearm in, because what the tricep would be doing is relaxing, right? So if I wanted to bring my arm in, I'm going to pull it in, right? And I'm pulling it in. I'm using the bicep muscle to do that. And the tricep muscle would be fighting against me if I was doing anything to activate the tricep muscle. So in this case, for that particular movement, the bicep is the agonist and the tricep would be the antagonist. So the agonist muscle needs to contract and it needs to be stimulated, but the antagonist muscle needs to relax, which means it needs to be inhibited. Now, conversely, if I want to do it the other, so if my arm is already like this and I want to bring my hand out, in this case, now it's the tricep muscle that's doing this work. And now the tricep muscle becomes the agonist muscle and the bicep needs to relax and become the antagonist muscle, right? And so Sherrington is looking at these groupings of two muscles that work together to control different movements at the joint. And what he found is that when the dog would flex the limb, the agonist muscle contracts, but the antagonist muscle is relaxing. He also noticed, by the way, and this was sort of a separate, partly related observation, is that the extensor muscle, which is working as the antagonist muscle for that particular limb, would relax, but on all the other limbs, the extensor muscles would actually contract. So you get this kind of weird opposite effect occurring in the other three of the dog's legs. So when Sherrington realized here that in order to create this flexion of the dog's leg, and you have to have the flexor muscle contract, this means that in order to make sure this works well, you would have an excitation of the flexor muscle, but what you also want to do is you want to have a simultaneous inhibition of the antagonist extensor muscle to allow this to work correctly, right? So that the joint does the desired thing. You don't want the agonist and antagonist muscles contracting at the same time. That would never really be a proper movement, right? You have to have one muscle active and the other inactive. So one muscle is excited and the other has to be inhibited, all right? So let's have that. Now we're going to call this an inhibition, an IPSP, right? An inhibitory postsynaptic potential. But how does it work, right? We already know how the EPSPs work because we covered that in the previous chapter, that the excitatory signal, when it stimulates a dendrite and a neuron, we're going to open up sodium channels, and that influx of sodium that's going to occur because of the electrical and the concentration gradient is going to raise the internal voltage of the cell, right? And that's going to get us, if we cross that threshold, we're going to trigger an action potential. But in IPSP instead, what it does is it opens potassium channels in the cell wall, right? What's going to happen with potassium? Well, because of the concentration gradient, potassium is going to leak out of the cell, and because potassium has a positive charge, that's going to lower the internal voltage. Something else that's going to happen is that it opens up chloride gates as well, right? And we haven't really said a whole lot about chloride in terms of anything that it might do at a synapse, at least not when we were talking about action potentials previously. But as it turns out, chloride plays an important role in the IPSP. Chloride primarily is sitting in the extracellular fluid. It is outside of the neuron. Chloride has a negative charge here. It's a negatively charged ion. And what's happening is that there is now a concentration gradient with more chloride outside compared to inside, right? And so now, just like potassium begins to leak out of the cell, which lowers the internal voltage, chloride is going to start entering into the cell, which will also lower the internal voltage. And so just as we saw in this graph, let's go way back here. Those two things together, either losing potassium or adding some chloride into the neuron, is going to lower our membrane potential below the original -70 to some other even further below zero number. And as we do this, one thing that we have to imagine is that this lowering of the membrane potential takes us further away from the firing threshold. So when we are at -70, which is a resting state, if we get some graded potentials in, we only need enough graded potentials to raise the internal voltage by 20 millivolts to go from -70 to -50, right? But if we have received an IPSP, that lowers the membrane potential down to say -80 in this case. I'm just going to call it that for now. Now we are 30 millivolts away from our firing potential. So we can still fire this neuron, but we have to have enough graded potentials, either through spatial or temporal summation, to raise the voltage by 30 millivolts instead of just 20, as we had before, right? So when the neuron's at a regular resting state, we have to raise it by 20 millivolts to cross that threshold. But when we are in an inhibited state because of an IPSP, now maybe we have to raise it by 30 millivolts, which means we have to have a stronger stimulus. So an inhibitory effect like this is not something that absolutely prevents the neuron from firing, but rather it's just making it more difficult. You need to have a stronger graded potential here, a stronger EPSP, in order to activate the neuron after the inhibitory signal has been sent to it, right? So that's what we are seeing here with the inhibitory discoveries, and here's the mechanism to explain it. And here is another picture of the neurons in the spine, but now there's a little more detail here because we can see what might be happening here when we apply that stimulus to the skin, like a painful stimulus to the skin of the dog. That's going to send that sensory signal into the spine. That's going to synapse onto the intrinsic neuron. That intrinsic neuron might send a signal to the brain so that the dog is consciously aware. But again, because this is a reflex, any conscious awareness does not actually play a role in controlling the reflexive flexion of the limb, but nevertheless, the dog can still, feel it, right, and be consciously aware, perhaps, of that. In separate research, for example, shows that these kinds of reflexive movements occur even if the connection between the spine and the brain is severed, right? So you can still see a reflex even in the absence of having any interaction with the brain. But this intrinsic neuron in the spine is simultaneously sending, as you can sort of see here, if you follow the lines there, an excitatory EPSP signal to the flexor muscle, or I should say the motor neuron that activates the flexor muscle. And then it is also sending a signal to another neuron, which is an inhibitory neuron. And that inhibitory neuron is connecting up to the neuron that controls the extensor muscle. And so that one intrinsic neuron is simultaneously exciting the flexor motor neuron, while through another neuron, it is inhibiting the extensor muscle. We can also imagine flipping this. So in case we wanted to do a different kind of limb movement where we want to excite the extensor muscle and inhibit the flexor muscle, there could be another set of synapses that are kind of overlapping on top of this or superimposed on top of this, where you get direct excitation of the motor neuron to the extensor, and then through the extra inhibitory connection, you end up with an inhibition of the motor neuron that controls the flexor muscle, right? Okay, let's stop it here, right? So as we have now covered a lot of the work that Sherrington had done, we're going to next move into the topic of what's really happening at the level of the synapse with all the neurotransmitters, but we also now want to discover or look at the history of the discovery of neurotransmitters, but we're going to do that in a second video.